Lee and Xie, Zhou, and Wang. My talk is benefiting from many discussion and also working opportunity with CIMMYT molecular breeding group. As many colleagues involved, and particularly, so we have Gary and Mike, and also Dr. Prasanna in the audience, and also thank Peter Windsor in the audience, and also Sarah Hing from the CD program. So we have been discussing a lot about the whole genome strategy. But I name it that way, but not everybody to talk about it. So for the funding, it's a lot of supporting to my activities in Mexico and also in China, particularly from the CIMMYT and the Bill Melinda Gates Foundation, and also several sources of Chinese funding. So I'm going to talk about 6 areas. First, they introduce some of the basic concepts. For the marker assisted selection, if you look at the markers, we have different definition. So you can have a single marker interval, haplotype, maybe also in the future for the heterological blocks, patterns, LD blocks, and also association profile. So we have different marker property that is for marker selection. For example, we have unique functional markers, neutral background markers, and also some markers significant, some are not significant. So I list here the 4 categories of the marker selection methodologies. So I call the marker-assisted gene pyramiding, backcrossing, recurrent selection, and also we heard a lot about the genomic selection. So for the whole marker-assisted breeding program, so usually we have several different platforms that is required. So the biggest one is the genotyping platform, and then phenotyping. And this is the phenotyping matter with all the environmental factors. With all this data generated, we need a breeding information platform, and also from all this process from natural or artificial crop population to the end, the lower gene plan developed. So we need various decision supporting tools. So what's the whole genome strategy? I defined as a set of the molecular marker-assisted breeding procedures that utilize the full genome sequence, genome-wide molecular markers to effectively address various genomic and environmental factors through a representative or complete set of the genetic resource and the breeding materials. This is long definition. So actually, it's the 3 important strategies that are now increased based on the understanding of genetic specific genomic regions, genes and new haplotypes, LD blocks, gene networks, and their contribution to specific phenotype. So I first present the strategy at the International Conference in 2010, and then we developed a manuscript called Whole Genome Strategy, published in the Molecular Breeding with my manager, Dr. Prasanna. So today, I'm going to share with you our update about this, the Whole Genome Strategy we call second edition. So basically, I'm going to cover up the factors affecting whole genome strategy. For example, we are now thinking more about the genome coverage. So that's all kinds of omics and also the sequence marker and haplotypes. Breeding populations, the whole profile for phenomics. So this is related to all the trait components, gene plasm representativeness, a precision of the phenotyping, and also the phenotyping protocols. In addition to this, we need to look at the G by E interaction and also the E-typing. That's a new word I coined for the environmental assay. Different selection method based on different criteria. And also we need the different tools to support our decision making. So because the schedule is really tight, I didn't get a chance to discuss with my group and also my director, Prasanna, here. So after my presentation, I hope you can give me comments and suggestions. We can finalize the second version of the strategy. First, I'm going to talk about genome coverage. We have heard a lot of story about the sequence and the resequence. If you look at all the first generation sequence project, there's a lot of genetic gap left, okay? It's because of three reasons. One, single genome that has been sequenced up to 80 to 90%, okay? It's not 100% for the genome genotype per se. So that are used as a reference genome, okay? Second, only one genotype has been sequenced with a relatively high resolution, usually representing a domestic variety, modern variety. So that's the problem you can say. So these sequence activities indicate that 20 to 50% of original reads from different ecotypes cannot be mapped to the reference genome. That's the biggest problem. So for maize, our resequence activity indicates that 50% of the reads cannot be mapped to the reference. And yesterday you heard from Peter about only 20% of slip from the landrace can be mapped to the reference. You see how big the gap we have. For rice example, just get it from Ken yesterday, you see 70% of the rice genome is indica, but our reference is japonica, okay? So you get about 15 to 20% of the sequence rate cannot be mapped, okay? So that's the two important clues we have for the story. Here I show you how the gaps are made. For example, if the wild relatives represent 100% of the genome coverage, okay? So maize landrace maybe just covered about 90% of the total. And the tropical maize may be another 20% lost. And then the temperate maize, a lot of 30% lost. But based on the 1B73, that's the temperate maize. The whole genome reference is only covered 90% of the B73. So you can see the big gap is left, okay? What happens if we use the partial genome coverage? So this is what I summarized here. Many times we get lost in map-based cloning. I have several examples from China. They do the rough mapping, but they cannot move to the fine mapping because no marker between the big gap, okay? Second, missing of the 40% and be more important culture if you do social mapping, which is the chip developed from the reference genome, okay? Biased estimation for many things, genetic diversity, population structure, LD, and also wrong haplotypes, what do you get? By the end, if you use some Marxist selection, it will be very ineffective, and also the results are not predictable. So, single genome-based reference provide only a partial genome coverage for a crop species. So we can say a lot of we call as a tenement bias. So we need a multiple genome-based reference for the whole genome strategy. Look at the maize. We have developed markers, different haplotypes from the marker SMP. So the first maize HEP map published in 2009. The second map, this is published in 2012. These two versions of the HEP map is really based on the reference genome, okay? Even we sequenced the hundreds of the inborn line, the unique sequence has not been used for the HEP map development and the chip development. So I have a sort of the 3 100x genome idea to represent the genetic diversity better. So the first 1,000 genome is the 1,000 genotype each at one time. That's the resequencing. So you get a much better representativeness. From those, you can get the reference genome, the loci, you can find the different alleles, okay, from different genotype. A lot of extreme, you sequence 100 times of the 10 genotypes, and then you get lots of the sequence data, and then you can line up, you build up the contig, okay. This contig cannot be mapped to the reference sometimes. So with these two data, so we can say maybe we can develop the third version of the HEP maps. So for example, the multiple genome-based HEP maps we can revolve the multiple lower near the loci covered by the single reference. At the same time, we can find the allele not covered by the single reference. So, and then we can develop the unbiased chip. So that's the stories going on with developing the HEP map 3. So over 800 lines used, and also trying to use the 83 deep sequence data from us. So, and also the GBS data, the Peter is talking about lots of inbred population that had been genotyped. So that's the HEP map 3 is going to be, or maybe 4. So when I talked to Alex, he said maybe a lot of version. So with that, if we use all the resequenced or GBS the population by linkage, we can build out different linkage maps. So put the markers in the missing region, okay? This is the gap. At the same time, the deep sequence data, we can build the contiguous. Some will drop in, in the gap. So with these two, we can combine these two together, okay? We fill the gap that way. Second, I'm going to talk about the market density and population size. I will go through this very quick. You see, the more marker you use, you can have the big impact on the maximum distance between markers and also average distance between the markers. So you can say we can do the mapping from rough to fine, even to the gene mapping, depending on how many markers you use. And also you see the population size will affect the maximum detectable distance and the minimum resolution you can get. That's the very small population size. But if you increase the population size to 1,000, you can see how big the effect will be impact on the association mapping. When we do the selective genotyping, we simulated population size from 200 to 3,000. You can say with the 3,000 individual, different tail size, you can see the co-tail factors vary from 1% to 20%. The mapping power can be, significantly improved with the larger population size. So lots of people talking about genotyping and phenotyping. So I'm just talking about the third typing. I have mentioned this idea in the International Crop Science Congress. That's come from the environmental assay record. So I think the E-typing should be treated equally as the genotyping and the phenotyping. So that's all the factors I believe we call external environmental factors including soil, air, light, temperature, water, and the cropping system, also the accompanying organisms. So there's lots of environmental factors. In addition to this, we can also define the internal environmental factors that come from the intercellular or intracellular. So they are contributed to various variation at a different level of omics. So if we put all the environmental factors together, this come from the internet and also the external, internal, external, including the GIS and also all these I just mentioned. So that's the GIS data we can use for the to divide the environmental site. So that's the we can use the e-typing to classify the soil property based on different strategy. So the key point is that someone mentioned yesterday, so I cannot get the name now. So it's important to have the environmental factors measured and rooming in. I call it rooming in to the plots and the individual plants. Look at that. We can really get this measurement to the each plot or each row. So that's a very good report from Japanese scientists. Use the environmental data and the genomics data to work together as the publishing in Cell. So that's the with that we can integrate phenotypical information and put the phenotype and the e-type information side by side and put together with all this GPE information. By the end, if you see, we have a very detailed information about the genotype. We put this genotype in a specific environment. We know all the environmental factors. I believe we can do a much better prediction of the phenotype. So selection method. So we have heard lots of marker seed selection method based on the for major gene, for background, for the whole genome. Okay, there's a different strategy for background, gene pyramiding, and the whole genome selection. So that's the protocol for the model development and also the population, breeding population selection based on the genomic selection. For this part, I'm going to focus on a little bit of the important topic. I think the key factors that are affecting genomic selection. That's including the number of markers and genome coverage I just mentioned, marker types, population structure, population size, the relationship between the training and the breeding population. If we have the known gene and the markers used, and also the precision, how precise the phenotyping, and also the inheritability of the traits. So this is all the factors affecting the genomic selection. So how to judge the selection effect? We have the genetic gain. Sorry. There's a basic concept to measure our selection response. So genetic gain, now we have experienced the concept development from the quantitative genetics to molecular breeding. So that's the formula to judge the genetic gain per cycle, per unit time. So that's including that gain, including genetic variance, heritability, selective intensity, and the cycle time. So for the genetic diversity, variance, so we just talking about how we can unlock the genetic variation from the original ecotype and also the wild relatives. So that's a different strategy. We can do that. Use single marker population structure variation and the haplotypes. Those is across different lines, across different genetic backgrounds. So we can define all this genetic variation. The second component, the factor is heritability. Actually, the estimation of the heritability can be improved through multiple trials and multiple environmental trials, multiple replications, and also this we can improve the heritability by control the environmental error. So we can control, if you have a much better environment control, you get a higher estimation of the heritability. Selection intensity, we can increase the selection intensity by increase the population size. So that's the factors, the strategies we can use to increase the population size. So all these strategies can be used in some way for different crops. Cycle time, that's an important one. It's the very big advantage for genomic selection. So before genomic selection, the international companies usually do 2 crops a year for maize. The one crop has to be phenotyped in the target environment as another in the off-season, okay? You cannot do 2 crops in the off-season because you're getting lost. Lots of desirable trail be getting lost. So with that, with the genomic selection, the company can do 2 off-season selection just based on the genotyping. So that's the very important advantage compared to, you can develop the variety in 6 years instead of 9 years. If you have so increase the shorter the cycle time. That's the, so I guess Prasanna is going to mention more about the market development application after me. So, so you can hear more from his story. The last session of my talk is the decision supporting system. We heard a lot of story about the data trilomy because of the different level of the data. We have a KB level, germplasm, and the MB level marked by accession, and the TB level of the omics data from different areas. So that's lots of data. With so many data, we have to integrate them in the platform. So some speakers have mentioned this. So put the genomic data and phenotypic data and environmental data into one platform. So with that, we need a breeding informatics system to handle all these information collection, integration, mining, and management. So that's so think about we can have 20 TB data. So sometimes we have problem to find a hard disk to hold all this data. So except that, the 3 key important for the breeding information database, as I just mentioned in the previous speaker about the ontology. I think it's also we need the important the session of the crop specimen and also cross crop database. And also this should be integrated with decision supporting tools. So that's the decision supporting tool list we need, from gene pattern evaluation, map construction, marker selection, even the IP protection. So from different data, GPE, and we have now many different tools, and then to generate a different output. It's all separate. And now we hope we have an integrated information system to do all these things, to do the one-stop shopping, to get everything done. So we have a pretty much a good expectation from the IBP system that's running by GCP program. I guess Jean Marcel is going to talk about this more. So with that, we hope the IBP will give us some complicate or package of the whole decision supporting tools. So this is the IBP tools under development for the different purpose. So by the end, so we have lots of challenges and opportunities. For example, we have the reduce the cost, also increase the scale and efficiency through high throughput genotyping, phenotyping, e-typing, and the information management and supporting system. With that, we can have a routine practice of marker plant breeding. So that means we have a GPE. I put another one, M, as a representative complete set of genetic breeding materials. Okay, so that's the whole genome strategy part. With that, we get all the desired part into our specific phenotype and the breeding product. So with the second edition of the strategy, we move the 3D profile of plant breeding to 4D profile. What do we get more? From this selection, we started from single phenotype-based selection, move to the multi-phenotype, and multi-phenotype by genotype, multi-phenotype cross-environmental by genotype, and then by the way, we have all this data across different time series. So that's we have the four-dimensional profile plant breeding. So Ben, thank you for your attention. Any questions?
Yeah. Please.
Yeah, I agree with measuring the environment, E-typing, but I cannot envisage how you integrate that in the current analysis system. Are you thinking of modeling or systems biology, or how do you integrate that?
That's a very good question. I've been talking with several quantitative geneticists. You know, the traditional quantitative genetics just handle the phenotypic data collected from the environment. And then they treat the environment as an E, okay? So you get a G by E interaction. So all the environmental factor is a mixture. So right now, we can have a component of each E, okay? Even measured for each plant by the end. So in this case, we have the G by E by each E interaction. So if you put the genotype in this specific environment, you get all these kind of E factors surrounding the plant. So that's why I'm asking the quantitative genetics to help me to develop the model with 3 dimensions, GPE together. Or you can predict the phenotype from the known genotype and the known environment. Those are 2 different stories. Very good question. A lot to do.
Manish?
Thanks, Dr. Xu, for this comprehensive presentation. And I'm really impressed with your concept for looking after in detailed e-typing, which you already you have mentioned in your PAC presentation in Genomics Assisted Breeding Workshop also. So that was very appreciative. Although yes, we have a challenge now how to integrate this data. It has been raised, and it is for the statistician and to help out us as a form in the form of decision support tools, because some part of it we have been using as an environment, but not all components which you have mentioned under e-typing. So this is very nice. I appreciate, and even I would like that in different platforms you keep on voicing it until acceptance.
Thank you.
Thank you.
Yeah, thanks. I think it's quite impressive, but I see one major problem. And the issue is that for, let's say, most phenotypes, you only have, well, let's say, for most genotypes, you have only one measurement on a number of plots at the end of the growing season. And for all your extra dimensions related to the environment, I mean, yes, it's true, in quantitative genetics, typically you only have an environmental main effect, and then there is a G by E term. But for the environment, you have only the number of degrees of freedom, let's say, for the number of trials that you did. So all these extra micro-environmental observations cannot be used in the modeling. And the only way around that is that you're going to take additional measurements during the growing season and even within the plot. But is that feasible?
Yeah, that's the key issue I mentioned. I call it room in to plot and individual. Otherwise, you have shared environment right across many plants. That's going to be
Okay, but I mean, I think
Not
it's
very-
nice to zoom in, but can we do that? I mean, do we have
There's
the
some-
means to do that?
Some protocols now, Prasanna can talk about more about. Some cases we can.
I'll await his presentation.
Okay. We see some phenotyping protocol and also the environmental collection, the data collection can go to the entry plot by plot. Yeah. Not many.
For certain traits, it is still possible to have time series. Of
It's not all.
For certain other phenotypes, yes, you are right. There could be just one measurement, but we cannot completely rule out taking, for example, severity scores based on weather patterns for important disease trait. You can have measurements of multiple series of phenotype across times. So it's a challenge, but for certain traits where environment microenvironment does influence a specific trait expression, it's indeed possible. One very prominent example in maize is the banded leaf and sheep blight, tremendously dependent on microenvironments.
Yeah, that's a challenge, yes.
Yes,
Actually,
please, the last question.
Oh, can I come back to the early part of the talk where you mentioned the problem of mapping reads onto the reference genome and the answer being to do more reference genomes. And so what's the decision process on what those 10 new reference genomes will be? That you're going to do a deep coverage of. So how do you decide what those are going to be? Because all the maze lines are very, very individual, right? So my question is, what's the decision process and is it really going to help significantly?
so actually it's two strategies. Simplest one, you do more reference genome, okay, if you have money. When I talked to Ada, she said for maize, because too much genetic diversity, we need sequence 10.
Yeah, but how do you
tropical
decide?
9,
Which
okay?
10? How do you decide?
At least. And then what I present here, if you don't have money for the reference, and then you can use all the resequence data and linkage data to find the markers in the gap, but it's not exactly the physical map. But you know the genetic map between these two markers. So that means by then maybe a lot of 10 years we have a much better reference, and then when we can map the marker back to the physical map. But at this moment, we can really improve the coverage of the chip for molecular reading or for gene discovery. So I don't know. So I'll see the program to do something for that.
Thank you, Ju. Thank you.
Thank you. Yeah.
I'm sure you have, many of you have questions for all the three speakers, but the session time is already over. I believe you can discuss those questions or points with the speakers during the coffee time. And with this we close the session. Let's give hand to all the three speakers once more.